All right, final video here of chapter 13 is going to have us delve a little bit deeper into this idea of genetic variation. And I talked about how important genetic variation is to the survival of a species, because it is this genetic variation that allows for a species to actually adapt, gain new kind of adaptations and new features that will help it survive in new and ever-changing environments. And so I had alluded to the fact that meiosis helps out with this genetic variation. We're going to talk about how that is. But one thing I'd like to point out here is that meiosis solely is not responsible for the genetic variation that we see in many, many species. The original source of all genetic diversity ultimately is mutations, okay? Changes to the organism's actual DNA sequences. And as far as I've ever heard, this is relatively random. Even if it is, not necessarily wanted, it is a random process, and sometimes you just don't know where these mutations are going to happen. Oftentimes mutations happen. They are really, really bad. They cause cancer, they cause disease, they kill the organism, okay? But sometimes these mutations happen in a part of the DNA that when the DNA is expressed, this new change in the DNA causes some kind of new trait to manifest. Now, we don't see anything as awesome as getting like laser eyes like the X-Men, or healing factors, or flying, or teleportation, or anything like that. We get, relatively innocuous things like blue eye color, or green eye color, or red hair, or, those are just some of the more visual ones that we see. But mutations do happen, and these mutations create different versions of genes that we already have. So oftentimes these mutations, these changes in the DNA sequences, happen in genes that are already there, and when they do happen, they change what the ultimate trait is going to look like when the gene is expressed, okay? Now, what meiosis does, and the way meiosis helps with this genetic variation that I've been talking about here, is that it takes these different versions of the genes and it reshuffles them into different combinations. And that sometimes is just as important in this grand scheme of natural selection and survival, because it puts together these different versions of genes that were possibly never seen before. And I will elaborate on that a little bit here, but it'll be more on the in-person side. I'm going to talk more in depth about how these reshuffling of genes can reshuffle and recombine different types of traits. Okay, so I want to get you guys to think back to the very beginning. One of the very first things I talked about here was the similarities and also the differences between siblings, but also the siblings and their parents. Okay. And I want you to keep that in the back of your head and remember that I said, yes, there are many similarities between you and your parents and what you look like and how your parents look. And you can also tell there's drastic differences as well. And so what we're going to get into here is how the genetic variation that I've kind of been discussing, this reshuffling of these genes, or these alleles as we call them, and these recombinations of these traits, how that leads to you as an individual never looking just like or exactly like identical to a parent. It is nearly impossible. Okay? And so there are three mechanisms that contribute to this genetic variation, and we've talked about at least one of them already. Okay? We've talked about crossing over already. We have seen independent assortment, although you have not been formally introduced to it. Now you are. Independent assortment, this is students, this is independent assortment. And so we'll talk in depth about independent assortment, and you may be very confused by that. Okay? So crossing over, independent assortment you've kind of seen already, and random fertilization is the random cherry to top off the entire genetic variation cupcake. Okay? So let's go ahead and delve into some of this. Independent assortment first, okay? Since it's the one you've seen in diagrams but haven't really known that you've seen it, let's go ahead and introduce it. I'm going to talk about it here, show a diagram, and then we're going to spend a more lengthy amount of time on it when I have you kind of in person, where you can sit there and ask questions, and I can confuse the crap out of you And hopefully you can jot things down and then ask questions after I've confused you, okay? Independent assortment is when homologous pairs of chromosomes orient randomly at metaphase 1 of meiosis. Okay, so this independent assortment effectively happens during meiosis 1. Okay? In independent assortment, each pair of chromosomes sorts maternal and paternal homologs into daughter cells independently of other pairs of homologous chromosomes. So right now I'm going to take a break. I'm going to go to a diagram, and I'm going to come back and hit these last 2 bullet points. Okay, so this is a diagram that illustrates how independent assortment kind of works, okay? And so we have to take into account probabilities here, all right, and randomness, as it were, okay? So let's use this possibility number 1 as kind of our baseline. If we have an organism that has 4 chromosomes, or 2 sets of 2, okay, right? So here's one set, and here's the other set. And this cell is going to go ahead, or this organism is going to go ahead and make some gametes, some sperm or egg. And we want to separate these chromosomes into their own little vessels, their own little sperm or egg, right? So the first thing we have to do is they will synapse. The homologous chromosomes will find one another as they have right here. The next step is that those synapsed chromosomes, homologs, will line up at the metaphase plate. Okay, so it turns out that they line up like this. The blue one here is on the left side, and the red one here is on the right side. If we look at the other set of chromosomes, the blue one here lines up on the left, and the red one here lines up on the right. Okay, great. And then everything else happens like we would expect it to. You would expect both of the blue ones to go here, and both of the red ones to go here. And then you'd expect the sister chromatids to separate like you see down here. Great. Okay, everything's golden. We love it, and we think it's great. Okay? Now, we have to remember that cells are 3-dimensional objects, right? They are spheres. And within these spheres, things can move in 3 dimensions. These things are not stuck to a table or stuck to a screen or stuck to a book page. They are spherical. They have movement even within the sphere, okay? And so what we have to kind of then think about is, okay, well, let's see, what if these chromosomes lined up differently at the metaphase plate, which is 100% possible. Let's say they lined up differently at the metaphase plate than the way they did in the first one, okay? So let's see if that is something that we can model. So we go ahead and have these guys synapse the same way that they would have before in prophase 1. This set of homologous chromosomes synapse together. During metaphase, they're going to line up at the metaphase plate. And we notice, okay, this blue one lined up at the left side the same way it did in the previous one. And the red one lined up at the right side the same way it did in the previous one. Okay, great. Everything's normal so far. Now we look at the smaller pair. Okay, and what we see is different, right? Instead of the blue one lined up on the left, the blue one's now on the right, and the red one is now on the left. And so what actually might actually happen is that, okay, if let's do this way. If this finger here is the red one in possibility number 1, and this finger here is the blue one in possibility. Oh, watch this. Hold on, I'm gonna go next level on this. That's a red marker. See that? Okay. I'm going to do the same thing in the back. My wife's going to wonder what the heck I've got blue and red marker all of my fingers for. It's for science. Not really for science teaching. Okay, that's done. All right, perfect. Now you can see it. All right, so the red one is on the right side, and the blue one is on the left side in possibility 1. Now let's say, just by happenstance, these things are floating around inside the cell, and let's say during metaphase they happen to just float and turn this way. Oh my, look at that. Cool, huh? And then they actually just turned around in space. And then during metaphase of possibility 2, we have the red one on the left side and the blue one on the right side instead. So it's just kind of flipped. And so what that's going to do is have consequences on what has actually occurred during the rest of meiosis. okay? And we get different combinations. So let's say there's, I think I'll leave it on that. But you can see that there's a different combination here. In this cell, there's a blue one and a red one, whereas previously there was a blue one and a blue one. And in this cell, there's a big red one and a small blue one, where previously we had a big red one and a small red one. And then in these gametes, we have further separation. Okay, so this is how independent assortment happens. Okay, these small chromosomes independently lined up at the metaphase plate from the big chromosomes. They did their own thing. They're free to float around however they want. Okay, they're not attached to the big ones. They're going to float and line up independently of all other chromosome pairs in the cell. Now, the reason I wanted to come back here is because that has implications on the different possibilities, of the different possible combinations or assortments of these chromosomes into the gametes based on how many chromosomes you have. So the number of combinations possible when chromosomes assort independently into gametes is 2 to the nth power. Okay? And so we think about, okay, what is n? N is like the chromosome number, the haploid number. Okay, so for humans, n equals 23. Okay, so for humans, n equals 23. So we can go ahead and plug in 23 here and calculate it, beep, bop, beep, bop, into your calculator. And 2 to the 23rd power equals 8 million possible combinations of chromosomes that can line up or that can be sorted into these different cells just by virtue of how these things will line up at the metaphase plate. Okay, so here we effectively only get 1, 2, 3, or we get 1, 2, 3, 4 different combinations. Okay, we get 4 different combinations, but that's because the haploid number of this is 2. 2 to the second power is 4, right? So that makes sense. Now 2 to the 23rd power, since we have n number of 23, is 8 million. So we would have 8 million different little, if we were to do this with our chromosomes, all color-coded and whatnot, we would have 8 million different little circles with different combinations of all of our 23 different chromosomes that we could possibly have. So that is going to, number one, add to the randomness. Okay, that's one thing that's going to add to genetic variation. Now we've got possible different combinations of genes that might be found on this chromosome that might be different from the ones that are found in the blue chromosome. Okay, crossing over. Crossing over produces recombinant chromosomes which combine genes inherited from each parent. Now, crossing over is 100% random. You have no clue if it's even going to happen between a homologous chromosome pair. Oftentimes it does. Sometimes there's multiple crossover events between the two arms of the chromosomes or the chromatids. Sometimes there's double crossover events on the same arm. Sometimes there's crossover events on one arm but not on the other arm. You just don't know. And so this happens on all of our 46 chromosomes, or all of our 23 pairs of chromosomes, and it's completely random. So what you're doing is you're putting a complete random shuffle into every single one of our chromosomes. Okay? And so crossing over, as I mentioned before, is a physical exchange of the material. And I had some diagrams in the previous videos that you couldn't really see crossing over happen, but here you actually can. It's a physical crossing over. Okay, so this sister chromatid from this blue one and this sister chromatid from this red one will physically move over and cross each other, and they call that the chiasma. Chiasma right here, where the arms actually will cross over one another, and then you will have a physical exchange of DNA. And so instead of all blue on this arm, we see a little chunk of red at the end. And instead of all blue on this arm, we see a chunk of red at the end. Instead of all red on this arm, we see a little chunk of blue. And the same thing down here. So this puts a completely random element into what's actually happening with combinations of genes or alleles. Okay? And so when we are together kind of in person, when I have you in person or online or wherever, I'm going to take this a little bit further. Okay, I'm going to take this a little bit further and make sure that you can understand what happens and how this causes different combinations of different versions of genes. And thus, we should be able to then understand how that leads to more randomness in our sperm or egg. Okay. Finally, we have random fertilization. Okay, random fertilization adds to genetic variation because any sperm, any of the sperm that a man makes can fuse with any unfertilized egg that a woman makes. And we have no clue what combination of genes it got in the sperm, and we have no clue what combination of genes it got in the egg, and then we have no clue which sperm is going to be possibly used to fertilize the egg, and we have no clue which egg is going to be used to make the baby. All of this stuff is random. Okay, it's more or less random which sperm makes it to the egg. It's more or less random which egg drops down into the uterus. We just don't know. Okay, so that's part of what happens in random fertilization, is that any random sperm that comes in can fertilize any random egg that was dropped down. Now, the fusion, that we can put a number to it. Okay, the fusion of gametes produced by a zygote with any of about 64 trillion diploid combinations. And so the way we get this number is if we take the 8 million possible combinations of chromosomes we got here, and we multiply, so like for one sperm, you'd have a 1 in 8 million chance to get a certain combination of chromosomes, and for any egg, you'd have a 1 in 8 million possibility to get any combination of chromosomes in that egg. And then you have whichever random sperm has this particular combination could come into contact with this random egg that has this combination. So the 1 in 8 million chance here and the 1 in 8 million chance here combined together, we multiply those probabilities and we get a 1 in 64 trillion chance of getting a certain combination of chromosomes. Okay? And then we add in crossing over, which you can't even quantify. Okay, 1 in 64 trillion chance of having a particular set of chromosomes that you and your parents have to make you identical to them is almost completely unlikely, right? But when you add in crossing over, which is completely random and we cannot quantify, and we don't even know if it's going to happen, but normally it does, but sometimes it happens multiple times, and sometimes maybe it happens one time or two times or three times. Oh my gosh, it's astronomical. You take all this into account with the fact that crossing over does happen, and the offspring will never be 100% identical to the parents. So this genetic variation that we see in the sperm and the egg is absolutely vital to keeping our population of human beings different from one another. If we're all the same, even if you're the same as your parents, that's disadvantageous to the long-term prospects of us as human species surviving in an ever-changing environment. Okay, if we're all the same and we continue to all be the same, that's great in an environment that never changes, but our environment's changing all the time. So this genetic variation keeps the adaptations around and keeps shuffling them around so that we can have new combinations of things maybe we've never seen before that are really, really cool. Okay? So, have a healthy respect for the fact that you do look like your parents because you are from them, but have a healthy respect and be appreciative that you are not exactly like them. I'm sure many of you can probably say, yes, I'm glad I'm not like my parents, but it is an important prospect for the human species in general. Okay, so it is the process of meiosis that does all of this for us, and I find that completely fascinating. So, jot down any questions that you have either over any of the 3 videos. Please bring them to my attention. We can talk about them. There's a lot of cool stuff we can talk about with this, especially with this final video. All right, I'll leave you guys to it. Have a good one.